Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our next talk, Evolution of Metastatic Melanoma, Insights from a Post-Mortem Study. I'm pleased to welcome Dr. Irene Lobon. Dr. Lobon is a bioinformatician researching cancer genomics. During her PhD at the University of Barcelona and the Institute of Evolutionary Biology, she studied somatic mutations in healthy brain and neurodegeneration. Dr. Lobon started her postdoctoral work at the Francis Crick Institute in London in 2020, where she studied the evolution of metastatic melanoma, especially under the selective pressure of immune checkpoint inhibitors. A warm welcome to you, Dr. Lobon. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Dr. Lobon's presentation. You can also benefit from some fantastic supporting literature, which you can download for free below the video player. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. And now, without further ado, I will hand over to Dr. Lobon to begin her presentation.
Well, thank you, George, for the kind introduction. And yes, indeed, I'm Irene. I'm a postdoc in the Cancer Dynamics Lab at the Crick, where we study tumor evolution, mostly renal cancer and melanoma. And today I will be talking about our insights from a recently published post-mortem study in metastatic melanoma. So melanoma is very common in the Northern European countries, Canada and Australia. And it results in almost 300,000 cases per year and about 60,000 deaths per year globally, mostly in younger people as well. At least with relates to, in contrast with other cancer types, it affects younger people, so it results in more years of life lost. And its incidence has increased over the last few years, almost 50%, and even more so in males. And the melanoma originates from the transformation of melanocytes that can then become invasive, leading to a local melanoma, and also leave the primary site, leading to metastasis. And sometimes the primary melanoma is not found, and only metastasis appears, and this is called melanoma of a non-primary. The idea is that in this case, the primary tumor was eliminated by the immune system, and only the metastases that were able to escape the immune system grew after. So melanoma that is in the primary stage is not very dangerous. So usually just resecting it is sufficient to cure the patients. But sometimes this is not the case, or patients will present directly with metastatic disease and will require further treatment. So just a couple of decades ago, the average survival of people in the metastatic state would be about 6 months. Then targeted therapy appeared, which just targets some common mutations in melanoma. And then patients went on to survive for about a year. But the treatment has really been revolutionized with the advent of immune checkpoint inhibitors. So here you can see a survival plot showing the percentage of patients that survive a long time. And as you can see now, thankfully, with the combination of 2 checkpoint inhibitors, over 50% of patients survive. And with more updated data, this is the case still, that about 50% of patients do survive. And what's interesting is that after about 3 years, you see that there's like a plateau of survival. So we think and hope that these patients are actually cured and they will not recur in their tumor type. But these treatments were only approved about a decade ago, or not even so. And so we'll need more time to see how these patients go on and whether they are actually cured or not. But of course, 50% of the patients do not benefit from this treatment. And currently, we don't have very good biomarkers to predict who's going to be in which 50%. So this is, again, a very useful and revolutionary therapy, but it is also quite toxic. So when you administer these drugs to patients, they can have consequences that can impact their life and even their survival. So we don't want to expose patients to a therapy that won't give them any benefit, obviously. So we need to become better at predicting who will respond and who won't respond. And hopefully also in that same endeavor, we can learn something about why some people respond and why others don't, and then understand the mechanisms of resistance and hopefully design new therapies. So what are immune checkpoints? So immune checkpoint inhibitors obviously inhibit immune checkpoints, whereas some pathways that the immune system has just so that it doesn't go overboard. Because the immune cells, so here you have a T cell, and this could be a tumor cell. And so what T cells do is obviously go and recognize with this MHC protein that is in the surface of all of cells. And it presents peptides from all the proteins within it to the outside, so that then the tumor cell can go and with a TCR see whether it is a match and whether it recognizes it or not. So T cells are not self-reactive mostly, for the most part. So many times these peptides are neoantigens or antigens that occur from new mutations. So tumors that have mutations will show them in their surface, and then T cells can recognize them. But the tricky part is that then if there's a lot of stimulation, the immune system will become inhibited through these immune checkpoint pathways. So what immune checkpoint inhibitors are is just these antibodies that will bind PD-1 or CTLA-4 that are some of these checkpoints, such that we can remove the brakes of the immune system. But for these therapies to work, the tumors need to be immunogenic, because it is only inhibiting these checkpoints. But again, this needs to be present for the T cells to recognize the tumor and then to kill the tumor cells. So melanoma is characterized by a very high number of mutations, or tumor mutational burden, TMB. So you can see here in a pan-cancer study where multiple tumor types were analyzed, and they showed the number of mutations per megabase. And you see that melanoma is the highest one. And it is characterized by these yellow type of mutations, which are C2T. And this is caused by UV signature. So different mutational processes will mutate specific nucleotides in different nucleotide contexts, so with different nucleotides that surround them. And that helps us understand which were the mutational processes that generated these mutations. And in the case of cutaneous melanoma, it's mostly UV, as you can see. And it results in very high tumor mutational burdens, about 40 per megabase, which is more than 100,000 per genome. So this is the reason why we think melanoma is immunogenic and why it responds so well generally to immune checkpoint inhibition. So here you see a plot comparing the TMB, so a number of mutations in different cancer types, with respect to their response to immune checkpoint inhibition, so one of them specifically. And you see that melanoma is here at the top, but importantly, you see that there's a relationship between these two metrics. So we think that the more mutations you have, so in this part here, the more likely it is that one of these mutations will be presented by the MHC in the surface of tumor cells, and then it will be recognized by the immune system so that it knows to attack the tumor. And also importantly, when looking at the number of subclonal mutations, and those are the mutations that are not in all the tumor cells, just in a few, melanoma cutaneous is the lowest. So the mutations that are present are acquired quite early through UV mutagenesis, and they are present in all cells. So if the immune system learns to recognize melanoma by a mutation, hopefully it will be present in all the tumor cells, and then they can all be killed. And just to point out that this is the reason why pan-cancer, the FDA has approved TMB as a biomarker for immunotherapy, because we think that if patients have a high TMB, their tumors have a high TMB, then they are more likely to respond. But all of this that we've been talking about is with melanoma of the cutaneous subtype, which is this one. But there are other types that are non-cutaneous, that are acral, which appears in the palms of the hand or the soles of the feet, mucosal in any mucosa, and uveal in the uvea of the eye. So the frequencies are very different by human populations. So in Europeans, cutaneous represents the vast majority of cases, but in people from ancestries of Asian or African origin, acral and mucosal can be more frequent. And they are biologically distinct. So TMB, as we were discussing, is very high in cutaneous melanoma from the UV signature, but this is not so much the case in the other subtypes. Most of them won't have a UV signature and they have a low TMB. And contrary, they have a very high burden of copy number alterations, so gains or losses of different genes or genomic regions that are early events, whereas melanoma, as early events, has very few copy number alterations, and these appear as the tumor diversifies later in its evolution. And this likely impacts the response to immunotherapy. So about 50% of patients have durable responses to immunotherapy, but this is much lower in other subtypes, especially uveal. And so there's a greater clinical need to understand the biology of these tumors and why they don't respond to immunotherapy and find new therapies. So recently, new molecules have been approved for other tumor types, including uveal. So hopefully in the next few years, we will have more therapies to treat these patients. So in our lab, we think that understanding cancer as an evolutionary process can help us understand better the mechanisms of resistance and the things that are selected along tumor evolution, and also understanding how repeatable they are across patients. And hopefully if we can look at a primary tumor or perhaps an early metastasis, be able to predict how the tumor is going to behave and whether the tumor will respond to treatment, so that we can decide when to give a therapy and which therapy will be more useful for different patients. So here you have a phylogenetic tree of one of the patients in our cohort as an example. And things that you have these different tumors that have different populations of tumor cells that are characterized by different mutations. So we can construct a phylogenetic tree in the same way that we will do that with species, for example. So if you have different primate species, humans, chimpanzees, you can look at how similar or different they are and build a tree from the differences, which will convey some information about their origin and the fact that they have common ancestors. Then we can map to these trees where different mutations appear so that we can understand whether they are early or late events and what that implies in terms of treatment. So some of the open questions in melanoma research and melanoma evolution are the tumor heterogeneity and organotropism, which means just that some features from the tumors will make them more likely to go to specific metastatic sites, to some organs where they can adapt. Which then what we like to think is that also these things have a high clinical relevance. So if we study them from an evolutionary perspective, we can hopefully help answering some questions in the clinic. So in terms of heterogeneity, this could be useful to explain discordant responses, which we see with immunotherapy. Some patients will receive immunotherapy when they have multiple metastatic sites, and some sites will regress and the tumors will shrink, but some other tumors will stay the same or keep growing. So we don't really understand how heterogeneous tumors are and what is causing these differences. And also, again, there's sometimes when patients have a single site that is escaping therapy and progressing. So we hope to understand this by comparing a lot of tumors within the same patient. Also, we don't really understand the routes of dissemination of the tumor cells and whether intermetastatic seeding happens. So this can inform surgical versus systemic interventions, because if you know that some metastases will seed others, perhaps removing that could be useful for the overall treatment of the patient. And also the timing of dissemination. So melanoma is one of the tumor types where after a primary tumor appears, many years can be in between that and metastatic disease appearing. So there's now some treatments that are given in the very early stage. And so we need to understand when is that the metastatic cells have seeded metastatic sites to see whether it makes sense to treat patients earlier on. And so to study all of this and to answer all of these questions, we've developed the PEACE autopsy study, which is a post-mortem sampling that occurs in multiple centers in the UK. And its chief investigator is at UCL, is Mariam Jamal Hanjani. Well, she organizes together with the team at UCL all the postmortems for the patients that kindly donate their bodies so that we can look at their tumors and understand tumor biology a bit better. And thankfully, we can obtain the clinical history and imaging from these patients so that we can track how different tumors have been growing and through all their therapy courses. And then again, we sample all the tumors so that we can better understand them. So in this case, I'm going to be talking about melanoma piece, which started recruiting at the Royal Marsden Hospital here in London in 2016. And more than 100 patients had thankfully enrolled, and 50 postmortems have already been performed. But we've now analyzed a pilot cohort of 14 patients. That is what I will be talking about today. So sorry for the gruesome pictures. I hope this is okay for everyone. This is just to highlight how metastatic disease can really spread everywhere in a patient's body. So here we see in a patient that had multiple tumors, and what we do is go and cut them all out so that we can see how heterogeneous they are. You can also see in brain tumors that are quite common in melanoma, sadly, but also we see regions of the same tumor that we can sample to see how heterogeneous they are within a single tumor. And again, we can see different phenotypes of the tumor as well, which is very helpful. For example, these ones that keep pigmentation versus others that maybe lose it. So we can then put together this clinical history and imaging, the histology of the tissues that we have pathologists review, and then we extract both RNA and DNA from these samples so that we can construct a picture of the tumor evolution within each patient. So this melanoma piece pilot cohort had 14 advanced melanoma patients that were from 4 histological subtypes. So we had 7 cutaneous, which you can see here. These are, each column is a sample, and then each block is a patient. So you can see we have 7 cutaneous patients, and then we also had 3 acral patients and 1 mucosal, and then 3 melanomas of a non-primary, or MUP, which is what I was explaining earlier, that not a primary tumor couldn't be found, but the patients presented with metastatic disease, and pathologists confirmed that this is melanoma. So these are all the samples we acquired, which are more than about 500 in total. So we've done whole exome and matched RNA for the most part from about 10 samples per patient, and then about 30 per patient of panel sequencing, so looking at melanoma-specific sites. And also we looked at single-cell whole genome sequencing in about 20 samples, which were mostly 1 per patient and a few others. So all patients were treated with immune checkpoint inhibition and other lines of treatment. So what we try to do is, again, understand the response and the evolution. And this work has been recently published in Cancer Discovery. So what we found was something that is expected. That is that we expect most SNVs, so most single nucleotide variants and indels, to be clonal. So you can see here the typical picture of a cutaneous melanoma that has a very long trunk of the tree. So many mutations that appear probably even before malignant transformation, just when in normal cells. And then short branches, where the different tumors diverge very little at the SNV level. But there's also different phenotypes. So for example, here we have a SAM-protected melanoma that doesn't have a UV signature, and it has an A gene signature instead. And we have also 2 patients that received chemotherapy as part of their treatment. And then you can see that they have much longer branches, and that this signature corresponds to chemotherapy signature. So this chemotherapy is generating new mutations independently in already existing tumors and making them more diverse. So what we wanted to understand, as I've said multiple times, is understanding how different are tumors in different metastatic sites across patients. And we actually found some differences. So for example, in this patient, we see that they had tumors in the brain and also some tumor in different locations in the thoracic cavity. And you can see from our phylogenetic tree that these were distinct. So there's one branch that seeds the brain and then one branch that seeds the other sides. And this is just a cladogram so that these branches can be better seen. And this is the same phylogenetic tree, just showing the proportion of mutations assigned to every signature for every node. And so you can see that these are very distinct tumor populations, probably reflecting the need of the tumor to adapt to the different environments. But other patients actually had a mixture of clones in the different sites. So here, hopefully you can see this with the asterisk is marking clones that are to brain only. And then the white one is marking clones that are both found in the brain and the thoracic cavity. And you can see that, and the others are only thoracic. So you can see that there's a mixture in most branches where clones are seen indistinctively. So perhaps it represents a more aggressive clone that for some reason is able to see everywhere a bit more easily. And indeed, we saw that the brain metastases were different in this cohort. So previous published cohorts of brain metastases that are from resected tumors, they represent perhaps a different phenotype of brain metastasis that people have reported has more copy number events. And actually what we saw in this cohort is that these tumors, some of them appear very late. So here you have for this patient, the tracking of the tumor size across time for different tumors. So you see, for example, the liver, the lung, and then in orange is the brain tumor. And what you can see is that the brain tumor was only detected at the last 2 scans. And earlier we could not identify a brain tumor whatsoever. But then, because we've sampled now all these tumors and some of them multi-regionally, we can reconstruct a quite accurate phylogenetic tree. And what we see is that the brain diverges as the first separation, the first branch that goes away from the rest of the tumor. So this means that in terms of the tumor cell population, the brain cells separated from the rest at an earlier time point. So these tumor cells diverge early, but then appear late. So this points to perhaps a clone that is not very fit, cannot grow very fast, and it is a distinct phenotype to the resected brain metastasis, and it points to some dormancy of these tumor cells. So perhaps they must have been there, it's just not growing at a pace that we could detect them. And again, this is just a study showing with single cell studies that they've looked at resected brain metastasis, and they found a higher burden of copy number events in these metastases. But when we look at this late emerging brain metastasis that can only be accessed through post-mortem sampling, you can see that we have a lower than expected burden of copy number events in the brain. We also saw the opposite trend in liver, but this was less clear. So this is something we want to explore further in the subsequent cohorts. And interestingly, we could find that for different patients, if we plot how distinct they are to other sites in terms of their copy number versus the time that it took them to appear with respect to other metastatic disease, you can see that there's a positive correlation. So you have some patients that are very distinct, meaning that probably they diverge early, but they appear very late, and others that are the other way around. So indeed, we see that there's a lot of copy number events generally in all these tumors. And we know this, that in melanoma evolution, I'll go to the next slide, that so melanoma, as we were talking about earlier, acquires point mutations quite early on. So when it is a melanoma, a primary tumor, and when it becomes invasive, it basically has all the mutations, most of them, that it will acquire. And so it is not that different along its evolution from a primary tumor to metastatic disease. But copy number alterations are appearing along the evolution. And indeed, in another post-mortem study, it was shown in 2021 that copy number alterations and whole genome doubling are more common in the late metastatic disease than earlier. So here you have, for different patients, they plotted different stages. So the primary in orange, then regional metastasis in gray, and in black distant metastasis. And you can see that whole genome doubling is more common at later stages. And when we looked at whole genome doubling specifically, which is when the whole genome is doubled and you have more copies of everything. So we found whole genome doubling in 14 patients, which you can see here. Again, these are columns for samples. And we've highlighted here whole genome doubling. So you are diploid tumor, it's here in white. 1 round of whole genome doubling is in light blue, and 2 rounds of whole genome doubling is in dark blue. And you can see that 11 patients out of the 14 had whole genome doubling. So it is a very common event, but we didn't found that it was necessary for all the patients. So some tumors didn't have whole genome doubling, and still the patient sadly died from the disease. So because we see this, it is so frequent, and also importantly, we found that 5 patients had parallel whole genome doubling, which means here you have an example, that when we construct the phylogeny, we see that whole genome doubling appears in multiple branches at multiple different points in the evolution of the tumor. And in this case, it was easier to assess that it was parallel events because not all the tumors are duplicated. But in some cases, even when all of the tumors are whole genome double, we know that there's a parallel event because we can time the SNVs to know whether they are pre or post whole genome doubling with a very simple assumption. That is that if you have 2 mutations and then you gain 1 copy of this chromosome, then you have 2 copies of this mutation that was there before. Whereas if now you acquire a new mutation, it will only be in 1 copy. So then by counting in how many copies are these mutations in, you can infer whether they were there before the whole genome doubling or if they appear after. So from this, we can sort of generate an ID for every whole genome doubling event based on the mutations that were there before. And what we find is that in these cases, we see whole genome doubling events that are different because the mutations that were there earlier are distinct. So this points us to some parallel events that probably highlight that this is an advantageous event for the tumor, but we don't really understand why this is advantageous for the tumor. So we tried to explore this further and have a bit more clues. So something we see a lot of is losses after whole genome doubling or together with whole genome doubling. Some of these we think happened before the event. So diploid tumors, you can see that the fraction of the genome that is gained or lost is not that high. And in many cases, it's more in the gains that the variance comes from. Whereas whole genome double tumors, we see have a lot of losses. And indeed, for most of them, the ploidy is 3. So for across the genome, you see mostly 3 copies of every genomic region. And this has been previously reported as well, that perhaps these losses happened before, and the whole genome doubling itself is a way to compensate for regions that are single copy of essential genes, mutations that accumulate and the tumor cannot escape. And also we saw that some of these regions that are lost are ones that have been reported by another paper before to be associated with no clinical benefit to immunotherapy, so with resistance to immunotherapy. So we think that at least some of these losses are functional and they associate with a lack of response to treatment in these patients. And indeed, we know that antigen presentation is a key pathway for these tumors to respond, because as I was explaining earlier, these tumors will have neoantigens that will be presented. So the proteins that do this presentation need to be there for then the T cells to recognize the neoantigens and kill the tumor cells. So it has been reported as a mechanism of acquired resistance that mutations in the beta2M gene that forms part of the MHC together with HLA can lead to resistance. And indeed, we saw a lot of loss of heterozygosity, meaning one of the alleles is completely lost. And in one patient, this was together with a mutation in this gene. And the frequency of this was higher than, say, in other cancer melanoma cohorts that are not exposed and such late stage. And we also saw a lot of LOH of other genes in this pathway, including JAK1 and JAK2. So we think probably this is contributing to the patient's, the tumor's resistant therapy. But this is not the only possible mechanism to explain why whole genome doubling, copy number alterations, and particularly losses are useful for the tumors. So one obvious strategy for the tumors is to delete the neoantigens. So basically, when after tumorigenesis, you will have certain mutations that are present only in a few of the tumor cells. And so if some of these mutations are very easy to recognize by the immune system, these cells will die leaving only those denoted here by the dark blue who don't have those very immunogenic mutations. And this is known as immunoediting, so that the mutations that survive this process will be less likely to be recognized by the immune system and will lead to immunosuppression or a lack of response to immunotherapy. Also, specific losses such as 9p21, which has been linked to CDKN2A and also a cluster of interferon genes, has been shown to alter the microenvironment. So here you can see in skin cutaneous melanoma, they classified the patterns of the immune cells around the infiltration. And so you can see that there's changes depending on whether this region 9p21 was wild type, was LOH, or completely lost. And so this we know alters the microenvironment. And also, and this is just what I was showing in the previous slide, is the alteration of the antigen presenting genes. So HLA, B2M, JAK1. So all of these proteins that are involved in this process, if they are altered or lost, at least partially, they may lead to less efficient tumor recognition and killing. So indeed, what we see is that our samples from this cohort have a colder microenvironment, so they are less infiltrated. What you can see here is one sample per column, and we've plotted together our PEACE samples, the RNA, together with the TCGA RNA samples. And we have information about whether they were treated or not, if it was immunotherapy and other types of therapy. So you can see that the TCGA are mostly untreated, but there are some that are treated. And then, that our melanoma these patients were obviously all treated with immunotherapy. So what we see is that what we did was to run a tool called consensus TME that tries to deconvolve from the bulk RNA the proportion of different immune cell types in the microenvironment, because you are sequencing them together with the tumor cells. And so you can infer, based on how we know they look transcriptomically, have an idea of how many of them you have. And what you can see is that the TCGA samples have much more higher levels of infiltration than generally the PEACE melanoma samples. There's some, that are a bit higher in certain like macrophages, which is something we want to explore further in our next cohorts. And indeed, you can see that it seems to be driven by probably treatment and also the stage of the tumors, because TCGA has a different immune score, which just recapitulates all of this, when they are treated versus the untreated ones. And PEACE has even a lower level of infiltration. So we think that the immune cells have been excluded from the microenvironment, and they are not there through some mechanism. So this is why tumors were not responding to immunotherapy. And indeed, it is known that chromosomal instability not only fuels tumor progression, but leads to immune escape and an immunosuppressive microenvironment. So this is work by the Bakum lab that has shown that chromosomal instability activates the SIG-STING pathway that is used by normal physiological cells to identify small fragments of DNA that are usually from viruses, so that then an immune response is activated at the beginning. So you see that there's a lot of interferon and like a lot of stimulation of the immune system. But when this acute stimulus that is supposed to be in the physiological state becomes chronic, then you have an immune suppressive effect. And the reason why this becomes chronic is because when you have chromosomal instability, which is defined by chromosomes breaking in cell division, and fragments of chromosomes just being there exposed to the cytosol, then this activates this pathway, but in a chronic manner, and this becomes immunosuppressive. And it has been shown that this can lead to chronic inflammation, which again is immunosuppressive and can favor metastasis. So we think that also just the mere chromosomal instability can be a reason to explain the lack of infiltration we found in these patients. And also in this cohort, what was nice is that because we have the information on the growth of specific tumor lesions, then we can map back to the tumors that were collected at post-mortem, we can compare within a single patient how are the tumors different, the tumors that responded versus not. Well, anything else is the same because it is the same patient, so the same systemic state, and a lot of the SNVs will be shared. So we wanted to compare how are the copy number events different in these tumors. So here you can see across the genome, a line showing the significance for gains and losses, so gains on top and losses in the bottom. And you can see that there are some differences, notably in chromosome 1 and chromosome 8, which are highlighted here below. And we see that there's this region where MYC and other neighboring genes are more frequently gained in non-responders. And this has been previously shown in published study to lead to an immunosuppressive microenvironment. So we think that these gains of MYC may be contributing to a lack of response in these specific lesions. So in conclusion, we see that there are multiple routes to lethality because we have some patients where we found parallel whole genome doubling events, whereas we found others that didn't have whole genome doubling. The TMBs were different across different patients, and still all the tumors resisted immunotherapy. Then we can see that polyclonal seeding leads to a lot of the intermetastatic heterogeneity. So we see that this organizes differently in different patients. Some of them have different tumors in brain versus other metastases, whereas other patients have a bit of a more mixed clones. And indeed, in some of the chemotherapy-treated patients, we could infer that some metastases were seeding other metastases, which I haven't talked about today, but it is all in the recently published paper. Then we also found this new phenotype of brain metastases that emerge late in the disease, but diverge early, pointing to dormancy, and a very different phenotype to the previously described one, which we want to explore further, so to see whether we can predict this in patients before when they are at an earlier stage, and we can act and decide treatment accordingly. Again, coming to the whole genome doubling, we know it facilitates chromosomal instability, a higher burden of copy number alterations, loss of heterozygosity of antigen-presenting genes, and potentially treatment resistance, but a mechanistic link needs to be uncovered. And we showed how looking at the heterogeneity within a single patient and comparing lesions that respond versus those that don't can identify new resistance candidates like MYC. And also we found that the late stage treatment metastases were very immune excluded. They didn't have a lot of infiltration, but we want to now understand a bit better the immune microenvironment and explore this further in the next cohorts. So with that, I want to thank obviously patients and their families. It is very generous on their part to donate these samples for our study. And then from our lab, the PI, Samra, and all the people that have worked with us, especially Lavinia and Desiree, who have contributed to this study tremendously. Also at UCL, we work with Kevin Litchfield lab and specifically with postdoc Alex Colton. From the Peace Consortium, obviously Mariam and Charlie Swanton, and Andrew Rowan, who attends all of the postmortems and helps coordinate all the samples. And then our collaborators for the single cell studies, and across all the the Royal Marsden and other hospitals, all the clinician and research nurses that really make this study possible. And in the Crick here, we've had a lot of help with our cluster and sequencing facilities. And obviously the funders, the MRA, the Crick, the Marsden, Cancer Research UK, and everyone else. So thank you.
Thank you so much, Irene. As with all the talks, we are likely going to have lots of questions from the audience, and I can see we have received some questions already, so I'll move straight on to the Q&A now. The first question is, how will we understand and target resistance to ICI if there is not a clear common resistance mechanisms across patients?
Hi, thank you. Thank you for the question and for organizing this. So I think that's quite a good question, that we didn't see in this study a common mechanism of resistance. So this is also in line with what others have found studying cancer samples, especially of patients treated with immunotherapy. So I think what is becoming increasingly clear is that there's multiple different mechanisms that are driving resistance in different patients. So that means that we will need much more data and more samples to be able to be powered to uncover all these mechanisms, and then have good annotation, especially clinical annotation, and to know the rounds of treatment, and then have a lot of multi-omic, hopefully, data, so that we know copy number events, but also the expression of genes. And then we can put it all together to try to understand responses. And actually, that's something we are doing currently in the lab. So we have other projects where we have samples before treatment with immunotherapy, and then we know their genomic landscape, and in some cases the expression of genes, and we try to see how can we predict responses to immunotherapy. And we think understanding responses or primary resistance will also help us understand the mechanisms in acquired resistance, which is in the case of these patients. So hopefully in the future we'll get some answers.
Thank you, Irene. Moving on to your next question. What could be the role of whole genome doubling in melanoma evolution?
Yes, so as I discussed in my talk, I think we see this very common event of whole genome doubling, and definitely it associates with more copy number events. And then we see that the copy number events are deleting genes that are involved in neoantigen presentation, and probably the neoantigens themselves, though we were not powered in the study to understand that. But then there's the whole axis of the immune suppression. So we think that probably the chromosomal instability is being immunosuppressive, and that's giving an advantage to the tumor. And besides this as well, is also other people like the Roger Law's lab at UCSF have shown recently that there's a lot of structural variants in these genomes. So they are more complex rearrangement that just gains or losses, and these have been shown to be very important for resistance to targeted therapy instead of immunotherapy. And then there's other people that have shown before that structural variants are also associated with a lack of response to immunotherapy. So we think that this chromosomal instability, creating structural variants, and perhaps also through immunosuppression somehow, are contributing to a lack of response to immunotherapy and the host's immune system. So an advantage for the tumors, definitely.
Thank you, Irene. It looks like we have some more time to answer a couple of more questions, which is, could CRISPR technology offer any leads to cure melanoma?
That's a very interesting questions. Yeah, there's definitely, I know of other labs have done CRISPR screens to first try to understand the biology. So when you have cell cultures, you can target different genes. So we see, as I was mentioning, these deletions, and we also see, for example, of a specific gene, CDKN2A, it's a tumor suppressor. So by doing screens, then you can definitely understand what is relevant or what is necessary for responses to immunotherapy. And then, I mean, once we know what is causing the disease, I think we would hope to be able to have some drugs that could target these pathways more than modify themselves, the cells. But then there's CAR T cells, which are T cells that are extracted from patients, and then they are engineered. So definitely all these engineering tools are very useful to create specific T cells that target the specific disease of the patients, that can be very helpful to cure melanoma.
Thank you. Moving on to your next question. Did chemo have an effect on WGD?
So that's a very interesting question. We only had, as I said, 3 patients with chemotherapy. And if my memory serves me well, I think they were all whole genome doubled, but we don't have power to say anything. I think we expect that the whole genome doubling from the trees precedes chemotherapy treatment. So I don't think we saw a correlation in our cohort, no.
Thank you. For the next question, which methods can be used to be more powered to study copy number changes in cancer evolution?
So as I was saying, with the structural variants, these things are very complex. So we think that long-read sequencing probably will be needed. So we're hoping to perhaps perform some nanopore on some of these samples. So then with very long reads, you can see the breakpoints and have a more accurate calling of these events that are completely blind to, in whole genome sequencing and definitely in whole exome sequencing.
Thank you. I have one final question for you, which is, are metastases seeded by other metastases?
Yes, so I was alluding to that at the end, that we saw only in the chemotherapy treated patients we could study this more accurately, because once you have the chemotherapy, this creates a very specific signature of mutations, which is like a timestamp on the tumors. So then we could see that some of these mutations that we know appear at some point, because it's when the treatment was given, then we could see that they were in multiple tumors. So we know that they must have appeared in somewhere. And then these tumor cells seeded multiple metastases. So definitely at the latest stages of disease, still some metastases seed other metastases, which is contributing to the overall tumor burden in patients, sadly.
Thank you so much, Irene. Unfortunately, that's all we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thank you, Irene, and everyone for your time today. I hope you've enjoyed it as much as we have, and we will be back tomorrow at 8 a.m. Pacific time with some more fantastic talks from our speakers and sponsors.
Thank you.
Should I approve that?